Proteins play a fundamental role in the functioning of the human body, facilitating various biological processes essential for survival. However, when these proteins misfold and aggregate, they can contribute to neurodegenerative diseases such as Alzheimer's and Parkinson's. Understanding how proteins behave and the role they play in these diseases has been a central focus of scientific research. Advances in microscopy and computational modeling have allowed researchers to study these processes in unprecedented detail, offering new insights into disease mechanisms and potential therapeutic strategies. Over a century ago, scientists began to identify key differences between healthy and diseased brains. The most striking difference observed was the significant shrinkage of brains affected by dementia, indicating substantial neuronal loss. This loss of brain volume correlates with the cognitive and motor impairments seen in patients with Alzheimer's and Parkinson's diseases. Researchers discovered that the brains of affected individuals contained abnormal clumps of protein, which appeared to be toxic. These aggregates, now known as amyloid plaques and tau tangles, were implicated in the disease process. Alzheimer's disease is primarily associated with two key proteins, amyloid-beta and tau. Amyloid-beta accumulates outside neurons, forming plaques that interfere with communication between brain cells. Tau, on the other hand, is primarily found inside neurons and plays a crucial role in stabilizing microtubules, which are structural components that help transport nutrients and signals within the cell. Under normal conditions, tau proteins bind to microtubules and support their function. However, when tau becomes hyperphosphorylated, it detaches from microtubules and aggregates into fibrils. These fibrils disrupt cellular function and eventually lead to neuron death, contributing to the cognitive decline characteristic of Alzheimer's disease. Scientific breakthroughs in structural biology have enabled researchers to study tau fibrils at an atomic level. Cryo-electron microscopy, cryo-EM, has provided detailed images of tau aggregates, revealing their structural variations across different neurodegenerative diseases. Although tau proteins misfold and aggregate in multiple conditions, the specific arrangements of these fibrils differ. This suggests that subtle differences in tau structure may influence disease progression and severity. By understanding these structural variations, researchers hope to develop targeted therapies that can prevent or reverse tau aggregation. One area of research that has gained significant attention is the use of supercomputers to simulate protein aggregation. Computational models allow scientists to observe how mutations in tau affect its ability to form fibrils. One notable mutation, known as P301L, has been found to accelerate the aggregation process, making it a valuable model for studying tau-related diseases. By understanding how specific mutations alter tau behavior, researchers can explore potential interventions that might slow or prevent disease progression. Experimental studies have further validated these computational findings. In laboratory settings, scientists have demonstrated that small segments of tau protein can act as seeds, promoting the aggregation of full-length tau molecules. This seeding mechanism is reminiscent of prion diseases, where misfolded proteins propagate from cell to cell, amplifying the disease process. Understanding the molecular basis of this propagation is crucial for developing therapies that can block or disrupt tau transmission in the brain. Another important aspect of tau pathology is its link to brain injuries. Repeated head trauma, such as that experienced by athletes in contact sports, has been associated with the accumulation of tau fibrils. Chronic traumatic encephalopathy, CTE, is a neurodegenerative condition characterized by tau aggregation in individuals who have suffered repeated head injuries. Interestingly, certain animals, such as woodpeckers and sheep, experience frequent head impacts yet do not develop tau-related diseases. Studying these animals may provide insights into protective mechanisms that could inform human treatments. The connection between protein aggregation and neurodegeneration extends beyond Alzheimer's disease. Parkinson's disease, another common neurodegenerative disorder, is characterized by the accumulation of a different protein called alpha-synuclein. Like tau and amyloid-beta, misfolded alpha-synuclein forms toxic aggregates known as Lewy bodies. These aggregates interfere with normal cellular function, leading to the progressive loss of dopamine-producing neurons in the brain. Dopamine is a neurotransmitter essential for movement control, and its depletion results in the characteristic motor symptoms of Parkinson's disease, such as tremors, rigidity, and bradykinesia, slowness of movement. Although Alzheimer's and Parkinson's diseases involve different proteins, they share common pathological mechanisms. In both conditions, protein aggregation leads to cellular dysfunction and neuron death. The spread of misfolded proteins from one brain region to another follows a prion-like mechanism, suggesting that halting protein propagation could be a viable therapeutic strategy. Researchers are exploring ways to develop small molecules, antibodies, and other interventions that can prevent or reverse protein aggregation. A promising area of research involves targeting the cellular machinery responsible for protein degradation. Cells have natural quality control mechanisms that help eliminate misfolded proteins. The ubiquitin proteasome system and autophagy pathways are responsible for breaking down damaged proteins and preventing their accumulation. In neurodegenerative diseases, these systems become impaired, allowing toxic protein aggregates to build up. Scientists are investigating drugs and gene therapies that can enhance these degradation pathways, potentially offering a way to clear harmful protein aggregates from the brain. Another avenue for potential treatment is immunotherapy. Researchers are developing antibodies that can specifically target Amyloid-Beta, tau, and Alpha-Synuclein aggregates. By tagging these proteins for removal, the immune system may be able to clear them before they cause significant damage. Early-stage clinical trials have shown promising results, though challenges remain in ensuring that these antibodies effectively penetrate the blood-brain barrier and reach their targets in the brain. Despite significant progress, there is still much to learn about the exact mechanisms that drive protein aggregation in neurodegeneration. The complexity of these diseases means that multiple factors, genetic, environmental, and lifestyle-related, likely contribute to their onset and progression. Studies have shown that genetic mutations can increase the risk of developing Alzheimer's and Parkinson's, but lifestyle factors such as diet, exercise, and cognitive engagement also play a crucial role. Additionally, environmental exposures, such as toxins and heavy metals, have been implicated in protein misfolding and neurodegeneration. Pesticides, air pollution, and industrial chemicals have all been linked to an increased risk of these diseases. Furthermore, genetic predisposition combined with environmental factors can create a cumulative effect, amplifying disease risk. Understanding these interactions is crucial for developing preventive strategies and personalized treatment approaches. Future research will likely focus on further unraveling the structural properties of misfolded proteins, identifying novel drug targets, and developing more effective therapeutic strategies. Advances in artificial intelligence and machine learning are expected to accelerate drug discovery efforts, allowing researchers to screen thousands of potential compounds more efficiently. As our understanding of protein misfolding and aggregation continues to grow, so too does the hope for effective treatments that can improve the lives of millions affected by neurodegenerative diseases.